Welcome to Talking Science. How's everybody today? Yeah? Last year, I think we had like a giant snowstorm on the morning or the day before Talking Science. Today, everybody should break out their suntan lotion. Who knows? So I'm Dr. Jeannie Garbarino, and I have the great pleasure of serving as Director of Rocky DU Science Outreach here at this wonderful institution, the Rockefeller University. By a show of hands, I'd love to see how many people have visited our campus before. Amazing. And so I would say about 60% of you, this is your first time. So I'm so glad to have you. Thank you for coming. And thank you to all of the teachers who coordinated. Let's give your teachers a round of applause, all right? Because sometimes it's teachers that really get us, all of us scientists who are here, it's the teachers who get us here. So I thank you from the bottom of my heart. And I will say, I love teachers very much. I'm even married to one, so. It is my privilege to be here this morning and to introduce today's speaker, Dr. Erich Jarvis. But first, I want to tell you a little bit about the history of the Rockefeller University. So just remember some history classes, take yourself back to the early 1900s, and think about how that lifestyle is different from what we might experience today, right? We didn't have refrigeration, there wasn't, the type of transportation was different, the limitations in getting in touch with people, there were so many different things going on, right? And one of the things that really plagued societies around that time and before is this concept of infectious diseases. It was really during the mid to late 1900s that we saw science really progress in our understanding of the pathologies involved in infectious diseases. And so it was infectious diseases that played a huge role in the start of this institution. So it all began in January 1901 when the beloved young grandson of John D. Rockefeller Sr., John Rockefeller McCormick was the child's name. He contracted scarlet fever and he died. And that was 2 months before this child's 4th birthday. The boy's death was a major blow to Mr. Rockefeller, and it reminded him of the limited resources available to prevent and treat the spread of disease. This personal tragedy inspired him to do something that had great significance for humanity. And at that time, almost 120 years ago, there were few medical schools that offered students opportunities for research, and no institution in the United States was devoted to studying the underlying causes of disease. And to make real progress, Mr. Rockefeller knew that scientists needed to understand the disease process at a very basic level. It wasn't just about treating patients. It was understanding why did they get sick in the first place? How can we prevent it? How can we treat it? So he founded the Rockefeller Institute for Medical Research, a dedicated biomedical research center that quickly became a leader in the fight against disease and in the effort to advance scientific knowledge. In 1955, the Rockefeller Institute expanded its mission to include doctoral education, and 10 years later became known as the Rockefeller University. Today, Rockefeller scientists conduct basic and clinical research, and their investigations touch on almost every disease or health condition you can think of. Some of their many accomplishments are listed on the back of your program. And as far as the university status goes, we are a university for graduate students. So we do not have undergraduates here. You cannot come to college here, but you would come here after you graduate from college to pursue a doctoral degree or perhaps a doctoral an MD degree combined with some of our partner schools. And so that means that but the coolest thing about getting a PhD in the life sciences here and elsewhere in the United States is how much you have to pay for tuition. Somebody throw out a number. How much do you think it costs to get a PhD? 50, I hear a 50,000. Let's hear another one. 60,000. Are we talking per year? Yeah? Okay, so 60 annually, 50 annually. Anybody else? 75, 80? Well, I'm gonna blow your mind right now. It costs $0 to get a PhD in the life sciences. Not only that, you can receive a stipend for pursuing a PhD, meaning you get paid to get a PhD. So I would be happy. Oh, well but that's just not here. This is what science is about. And for anybody who's interested in pursuing that in the future, come find me during the lunch hour. I'd be happy to talk to you about that. So here at Rockefeller, we have 200 graduate students on campus each year, working with more than 600 scientists who range from postdoctoral fellows to the professors who head the university's nearly 80 laboratories. And over the years, the importance of the discoveries made at Rockefeller has been recognized throughout the world. 25 Rockefeller scientists have been awarded the Nobel Prize. That is more than most countries regarding their Nobel Prizes. And 23 have received the Lasker Award, which is also known as the American Nobel. So how did I get here? Well, I was a postdoc here at Rockefeller after doing my Ph.D. at Columbia in metabolic biology. But I still every day walk onto this campus and I think to myself, wow, I am so lucky to be here. And I'm so lucky to be able to continually welcome students like you through these doors and ensure that everybody can have access to the process of science in the authentic way that science is done. And so when I was in my postdoc, I pitched that we need to do more for the high school communities, the middle school communities, teacher communities. And they were like, okay. And here I am, 8 years later. And that's because we believe that science for the benefit of humanity, which is Rockefeller's mission, also includes sharing our science with students and teachers. In fact, we work year-round through RockEDU to present fun, authentic science experiences to thousands of students across the city, whether through our science cafe, so RockEDU Presents, which happens every month and is hosted in one of our other auditoriums. We have mentored research programs, so we have Lab Jumpstart and the Summer Science Research Program. Anybody here, we have a couple of SSRP and Lab Jumpstart folks here. Anybody applying for the SSRP, feel free to come talk to me during the lunch. We can chat about it. And one event I especially look forward to every year is this event, Talking Science. This is a very special part of a Rockefeller tradition where scientists give back. This lecture series began in 1959 when it was presented as an annual Christmas, then holiday lecture on science for high school students. It was established by Alfred E. Mirsky, who's a biochemist and is one of the pioneers in unlocking the secrets of the cell nucleus. That sounds pretty cool. Dr. Mirsky modeled the lecture on a popular series of science talks for young people established in London in 1825 by Michael Faraday, who is considered one of the greatest experimenters in the history of science. Fun fact about the Faraday lecture series, I read somewhere, and I have to dig up where the citation is, but I read that because his public talks were so popular that they established for the first time the concept of one-way roads so that people could attend the lecture series and then people leaving could go through another road. So the concept of one-way roads, I need to fact check and find the citation, but I read somewhere that is the case, and I'd love to attribute it to Michael Faraday, but we'll see. Today we are pleased to welcome students from more than 50 schools from across the 5 boroughs of New York and beyond. So thank you again for spending your Saturday with us in pursuit of scientific knowledge. So I love seeing this. And on behalf of the university, I would like to gratefully acknowledge Merck and the American Museum of Natural History for their generous contributions in helping to present today's exciting program. I would also like to recognize Dr. Raphael Berger, Associate Principal Scientist in Chemical Biology with Merck, who is here with us today. Thank you so much for joining us. So thank you to American Museum of Natural History, to Merck, and to Dr. Berger. So let's give them a round of applause. I'm also so excited and so grateful to have Rockefeller President Rick Lifton and Deb Lifton in the audience with us today. Thank you so much for sharing your Saturday with us. And now I'm going to introduce the man of the hour. It is my pleasure to introduce Dr. Erich Jarvis. Erich was born, oh yes, let's do it. Erich was born in Harlem, though we did discuss you did grow up in the Bronx, which is also my borough. So let's hear it for the Bronx folk. Yes, the Bronx is amazing, and was raised by a single mother. And despite challenges faced growing up in a poor family and in a tough neighborhood, Eric's mom knew the importance of a good education and encouraged him to strive for excellence in school. She promised that if he worked hard, he could make a difference in the world. Eric was and is a very hard worker, and he's clearly making a difference in the world. His first passion was dance. He attended the High School of Performing Arts in New York City, and you may have seen the movie, or maybe you've heard your parents talk about the sitcom Fame. I loved that show growing up, which was set at his high school. And if you haven't seen it, you should go check it out. Especially, I love the fact that you can just like go on YouTube or go on Netflix and find all of these sort of historical sitcoms, but they were amazing. And he trained at the Joffrey Ballet School and danced professionally with the Westchester Ballet Company. He was on his way to becoming a professional dancer, and I have definitely seen his moves in some of our dance parties here at Rockefeller, and it's show-stopping, absolutely show-stopping. But dance wasn't Eric's only interest. He also had a love for science. As high school was coming to a close, he was faced with two options: chasing a dream of a dance career or pursuing science. Eric decided that ultimately he would have a better chance at making a difference in the world as a scientist. So he enrolled in Hunter College for undergrad, which is a CUNY school just 3 blocks away from here. And while he was there, he worked as a Minority Access to Research Careers Fellow, sponsored by a government organization that provides research training opportunities in biomedical research and sciences for underrepresented minority groups. And he received a bachelor's degree in mathematics and biology. He then enrolled here at Rockefeller University, becoming one of the few African-American students accepted in the history of the university. He went to earn his Ph.D. at Rockefeller in molecular neurobiology and animal behavior after researching vocal learning in songbirds, the model organism for his current investigations. After spending 16 years on the faculty at Duke University, in 2016, Eric returned to the Rockefeller University, where he is now a professor and head of laboratory of Neurogenetics and Language, and is the university's first African-American professor. And I think that definitely shows that science has a long way to go. An investigator of the Howard Hughes Medical Institute since 2008, Eric is the recipient of numerous awards, including the National Science Foundation's Alan T. Waterman Award, an NIH Director's Pioneer Award, the 2015 Ernest Everett Just Award from the American Society for Cell Biology, and 2019 NIH Director's Transformative Research Award. He's also a member of the Hunter College Alumni Hall of Fame. Eric's research grows out of his long-time fascination with why some animals, including humans, are vocal learners. They are able to hear, learn, and repeat sounds, while most others do not have this capability. In the first half of today's talk, Birdsong and Human Language, Eric will explore the difference between vocal learners and non-vocal learners, and he will delve into the similarities between human and songbird neural networks. And he will share exciting innovations and discoveries that have emerged from research conducted in his laboratory here. After the lunch break, in a talk called Learning to Fly, Becoming a Scientist in a Diverse Environment, he will go into more depth on the challenges he faced on his personal journey to becoming a scientist. And now, without further ado, let's welcome Dr. Erich Jarvis to the podium.
Thank you very much. I appreciate it. Of course. Let's do it. Well, thank you, Jeannie, for that wonderful introduction. When I listen to it, I'm like, who is she talking about? And I'm so happy to be here with you today and to talk about science as well as becoming a scientist. And so I'm happy to see that there's a new generation of students who are interested in what we're doing and interested in maybe becoming scientists yourself, or at least applying scientist teachings to everyday life. I tell you, we're going to need it as the world is changing. So, yes, I'm going to talk to you about two things today. The first part is science, and then the afternoon we'll talk about some things that I've learned in my journey of becoming a scientist, both professionally and personally. So my favorite topic is understanding how the brain works. how the brain controls complex behaviors. And one of the behaviors that I'm most interested in is language. And the reason why I chose language is because when I was in junior high school, and also my mother taught me, is that if you learn to play a hard instrument, then easier instruments are better. You're better at it. And I thought, well, language was a hard instrument. So if we learn how the brain controls language, then we can figure out everything else about the brain. And so, but it turns out we humans are not the only ones that have this ability to do what I'm doing now, this ability to produce learned imitated sounds. Songbirds do as well as some other species that I have up here in this slide, including Jennifer Lopez. All right? And so what I'm going to tell you about in the next hour or so is really how have we understood this behavior, that's what we're going to call it, at three levels. That is the actual behavior itself, the brain anatomy involved, the actual genes and the genome involved in it, and then testing some ideas based upon these three levels of analysis in mice. All right? So let's get to the first one, vocal learning, and a component of spoken language. When I say language, and I'm going to say particularly spoken language, many people don't realize that it actually has multiple components of different kinds of behavior that give rise to what we're doing now. And some of those are more ubiquitous among species, like what we call auditory learning, or syntax, the ability to understand sequences of sounds. And some are more rare, like vocal production learning, or speech and song, the ability to really produce those sounds. And so what I'm going to say to start off here is spoken language is a specialized form of learned communication, vocal communication that is, with some forms or components found in most vertebrates, and you'll see that in a minute, but a highly specialized component found in us in just a few species. And when I say spoken language, I'm going to say it's equivalent to speech. So this specialized component, we're calling vocal production learning. 5 groups of mammals have it. Us humans, cetaceans, those are whales and dolphins, bats, elephants, and seals. What group does seals belong to? Does anybody study classification in their biology classes? I'm going to do some trick questions. So it starts with a P. Seals and sea lions and so forth. Okay, that's all right. It's called, there you go, pinnipeds. Thank you. All right. And there are 3 groups of birds that have this ability. The parrots, many of you know about parrots, can imitate sounds, but also songbirds and even the tiniest of birds, hummingbirds. Now, this is different than the ability of perceiving spoken language. That is auditory comprehension learning. Actually, many animals have this, like your pet animals. You can teach the dog to understand the word sit, right? Or siéntese. What language is that? In Spanish. Usuari. What language is that? Does anybody know? Japanese. Yeah, Usuari means sit. So I remember one time I actually grew up in the Bronx and Queens, and I was taking a friend back to visit the old house I grew up in Queens, and there was a dog standing at the gate and started barking at me, and I was like, I didn't want to alert the neighbors that a former child that lived in this house in Queens is now stalking them, right? And I'm telling the dog, sit, be quiet, be quiet. And finally I said, siéntese, and the dog sat, okay? That's auditory learning, all right? But a dog can't say, okay, you got it, I will sit for you, okay? What a dog will do is woof. And you can teach a dog to produce different kind of woof sounds for food, for petting, and so forth, but a dog won't actually say, pet me. And so, so what we call that is vocal usage learning, when a dog learns how to produce innate sounds in different contexts, but not vocal production learning. Now, not all vocal learners are equal. Besides humans, parrots are the most advanced, and here's an example of a parrot, a budgerigar, which is a small parakeet, basically parakeets are small parrots, that was raised with humans for 4 years and can produce roughly 400 human words. And here's an example of some conversation he's having.
Never shake a baby bird. That would surely be absurd.
Never shake a baby bird. That would surely be absurd. Are you not a crook? My name is Tesco. The cat, meow, meow.
Yeah, so disco is impressive, right? Imagine trying to get that out of a dog or a chimpanzee or something like that. And what's going on here is disco has learned a whole bunch of human, in this case, English words, and about 75% of the time, disco seems to ramble on with these different words, even with parrot vocalizations itself, in an incomprehensible manner, and sometimes comprehensible, like when Disco saw the dog and said meow, kind of got the category a little correct, but the actual individual animal wrong. But a dog is not going to go meow. And so, so this really excites us. What's going on here, right? Every once in a while, and can we have the volume up a little bit for this? Every once in a while, a study comes out where one of our closest relatives, in this case a gorilla, also raised by humans for many years, produces some type of vocalizations or sounds that get scientists excited, that says it's getting close to humans, but not quite. And here's an example.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
Yeah, so Coco is doing excellent here, but Coco can't say, my name is Coco, I'm a gorilla, not a spy, or something like this, okay? And what's going on here is that Coco has very good, decent at least, auditory learning. Coco understood all those human speech sounds that this person asked. And Coco responded to the question asked and actually made a cough. But Coco can't produce more novel sounds beyond a cough and gorilla innate calls. And so Coco has good auditory learning, but not good vocal production learning. She's voluntarily blowing air through the vocal tract, but not able to modulate that air to make the kind of sounds that I'm doing now and when you're laughing. All right. Actually, gorillas can laugh. So, so that's caused us scientists, ever since I was a student, and now in my own lab, to ask this question: why can we humans and these species of parrots and other birds so far distant from us by 300 million years of evolution can do things like imitate these sounds like apple when we're trying to get our closest relatives to say it, and they can't, right? And the parrots will go even further and say golden delicious, dummy, And so, so what's going on here? Is it in the brain? Is it in the organs? Is it in the genes? And so forth. And even many attempts to raise a monkey or an ape in a household for many years can't get them to imitate sounds. So I argue it's in the brain anatomy. And I'm going to tell you now for the second part of my talk, what is it about our brains and the brains of these birds that allows us to do this that others can't, and how did that come about over evolution? All right, so actually the initial discoveries were here at Rockefeller. My Ph.D. advisor discovered in the brain of songbirds a set of brain regions that control the ability to vocalize not found in others. And when I was working with him, Fernando Nadaban, what we found is as follows, is that when canaries produce their learned song, this is a songbird. Now, it's not imitated speech, but it's canary song. It's learned. And when they produce that, what happens is that there are genes in the brain that have their gene products turn on as a result of the activity occurring, the firing. Has anybody heard of action potentials? Okay, good. Has anybody heard of messenger RNA? Okay, good. Your teachers are good to you. All right. And so what's happening is that the action potentials in the brain that are controlling these vocalizations are turning on these genes in this white signal here, the messenger RNA for it, after the birds sing. And we find that the more a bird sings in a half-hour period, the more increase of this messenger RNA product of these activity-dependent genes that are occurring. And so what this means, this is a slope of 1, okay, in this curve. What that means is that for every song the bird sang, we get a 1-fold increase in the messenger RNA of this gene in this half-hour period. It's pretty robust. And so, and we can also see other brain areas light up when the bird is hearing sound and so forth. And so what we did is then went out and tested this kind of behavior on many bird species, and we're also sequenced the genome of many bird species, all of these species here, but not, we didn't test all of them, but many of them, where we looked at songbirds, parrots, and hummingbirds, and their closest relatives, according to different publications out there, is who their closest relatives. And what we see here, looking at their genes, we can actually get what we call a family tree of birds. And the first thing we notice in green here are the vocal learning birds, the songbirds, the parrots, and the hummingbirds. I also put the dot there as to when we think vocal learning evolved in them. So they indicate 3 independent gains of this trait, because all these other species in between are thought to only produce innate sounds like cocoa, okay? Including penguins, including eagles or owls and so forth. And so another possibility is, well, maybe they inherited from a common ancestor back here. I'll get to that in a minute. Back here at the time of the extinction of dinosaurs. But that would mean then there have to be 10 losses to occur over this 60 million year window of time. It's like humans and chimpanzees and Coco's ancestors having the ability to produce speech, and then we lose it. And so why would that be so remarkable? When we looked at the brains, using that gene activity thing that I just showed you in the last slide, when we look at the brains of the vocal learning birds and the non-learning birds, what we find is as follows. In all species, in blue here, we have regions that are activated when they hear sounds, like what's happening to your brain as you're hearing me. All right, this is called the auditory pathway. We can find the auditory pathway in the forebrain of all of them, consistent with all of them having auditory learning. But only the vocal learners had brain regions in their forebrain, colored in red and yellow here, that you cannot find in the non-learning species. In red are the pathways involved in the actual imitation of the sound. And in yellow are the pathways that go down to the neurons that control the muscles for vocalization involved in producing the imitated sounds. And so what's happening is auditory information is feeding into the red pathway, telling the yellow pathway what to do on these gray regions down here, which are the brain regions producing the chicken innate crows or Coco's innate vocalizations. So what happened here? In the last 65 million years, it looks like a very similar pathway evolved 3 times, okay? Quite remarkable, with 7 brain regions in similar connectivity. It's like the evolution of wings evolved multiple times in birds and bats, and each time it was something similar. How could that have happened? And if it evolved similarly 3 times in birds, well, what about us humans? Are we any different? And so we began to ask this question. And here is a songbird brain, looking at it from the outside. And here is a human brain. And here is a songbird brain, in particular a zebrafinch, to scale with the human brain. All right? You can fit 3,000 zebrafinch brains into a human brain. So what does this mean in terms of this trait? Size doesn't matter. That's right. Neither does cortical folding matter. You got all this surface area to control lots of different behaviors and a smooth cortex here. But yet you can have this trait in both of these kinds of brains. What matters, we think, might be what's going on inside the brain. But to get at understanding what's going on inside the brain, we had to evaluate a century-old view about how bird brains are related to human brains. At the end of the 1800s, beginning of the 1900s, the patriarchs of comparative anatomy took Darwin's view of evolution, Aristotle's view of scala natura, and their own religious and prejudices views, and proposed the following, that in the human brain, in purple here, you can find these regions that are going back to different ancestral states of a linear scale going from primitive to more derived forms in different animals, such that at the bottom of the human brain, or the bird brain here, is what they call the paleostratum, what fish had. They passed that on to amphibians, and they called it the neostriatum, who then passed it on to reptiles. They called it archae for old striatum, and so forth. And the bird brain was thought to be one large, what we call basal ganglia, or primitive structures. And only in mammals evolved this latest and greatest achievement they called the neocortex, for being new. All right? and that you couldn't find hardly any neocortex in any of the other vertebrate species except for mammals. And in humans, it really greatly expanded, and it expanded best and biggest in Europeans, next in Asians, and smallest in Africans, and even smaller in monkeys. And that's where we get the term neocortex from. It was totally false, all right? I'm proof that it's false, all right? And but it is a case of how culture and beliefs influence science, so be careful. All right, so I organize a group of scientists from all around the world with experts in brain anatomy and behavior, and we evaluated all the evidence over the last 100 years, as well as obtained more evidence ourselves, and came up with this, what we call Martin View. This is really the organization where the bird brain and other vertebrates as well have a large cortical territory. It's just not layered as it is in mammals, it's clustered. And here is the true basal ganglia, and the basal ganglia is not just involved in innate behaviors. It's actually impacted in Parkinson's disease. It involves learning how to do many kinds of behaviors. And so with this organization now better understood, actually when we published this, some of the news media said bird brains should no longer be stupid and tell Webster Dictionary to change their definitions and so forth. And it should not be considered that way. So now with this Martin view, we can compare these birds to humans. Remember this diagram I showed you? Well, this is now what we know about human brain organization compared to a non-human primate like Coco. And what we see is that humans and non-human primates and other mammals have an auditory forebrain, whether they're a learner or not, just like birds, all right? But only the vocal learning, now in this case humans, have brain regions that are responsible, like Broca's area, for imitating sounds, or laryngeal motor cortex for producing the imitated sounds. And that's quite remarkable. There are parallels in their functions between us humans and these vocal learning birds. And when we proposed these two ideas for birds and between birds and humans, some religious groups got a hold of our results and actually put them in some online text to say this helps prove the existence of God, because how could this have come about over the last 300 million years independently like this? And we really didn't have an answer to how could the human brain be similar to birds more than it is to a non-human primate, and non-human primates be more similar to chickens, all right? So even in terms of the connectivity of a direct connection from the forebrain down to the neurons that control vocalizations. And I didn't have an answer until we came up with this discovery here. We found that not only the brain regions active in vocalization showed this activity-dependent genes in response to the action potentials coming on, the messenger RNA here, but the brain regions around those song learning nuclei that control vocalizations are also active when animals are learning how to do different kinds of behaviors, like walking and so forth, all right? Or flapping their wings. And all of these regions are directly around the 7 song nuclei we see in the brain involved in vocal learning, except these aren't vocal learning brain areas. They control other muscle groups that control limb muscles and so forth. And the connectivity of these song nuclei for vocalizations, like these white arrows here, are similar to the connectivity for the brain areas that are controlling movements for other body parts. And we found this was the case for songbirds in this image, for hummingbirds, for parrots. And in the non-vocal learning species, what did you think we found? Did we find these? No, we did not. But we found these regions here. Okay, so we found the movement areas that control other body parts, but not for the vocalizations in the forebrain. And so, and guess what? At around that time, a colleague of mine, Stephen Brown, had done some similar experiments with humans, in this case, getting dancers to do choreographed sequence movements while having their brains imaged for activity, and found that the brain areas in red here showing the highest activation are directly adjacent to the brain areas that control speech. Okay, so, and around also that same time, after we had discovered these brain areas controlling learned movements in the songbirds and parrots, it was discovered that only vocal learning species can learn how to dance. And here is an example of a cockatoo, which is a parrot.
Snowball. So, Irina, this is so much better than Dancing with the Stars. Tell us very briefly what Snowball can do, and then we'll witness it for ourselves.
Well, Snowball can dance to a beat, and he actually adjusts his dancing to fit the tempo of music.
Shall
That's to the downbeats of the music. I want you to pay attention to that. And he does it listening to sound, but also observing people.
Does he like for everybody to join in? Shall we?
Oh, by the way, these are a bunch of scientists. One from Rockefeller, another one who used to be at Rockefeller. And see, are the scientists more on beat or the bird? Okay. So since this discovery of Snowball, there have been now many discoveries of many parrots out there and some songbirds that actually can learn how to dance rhythmically to the beat of music. And so when they try to get this to occur in a pigeon or a chimpanzee or some other species that doesn't have vocal learning abilities, or dogs, dogs will actually respond to human music. They'll start jumping up and down with you as you're dancing. But dogs have very difficult time to actually keep the rhythm of the beat. I know some humans do, but you're still better than your dog, okay? So putting all that together, we came up with what we're calling the motor theory of vocal learning origin. Whether this be a bird brain or a mammal brain, that all vertebrates have this innate brainstem structures here in the midbrain and the medulla that control the production of innate sounds like the dog barking, the chicken crowing. And in their forebrain, all these vertebrate species have a pathway involved in learning how to produce movements and the pathway that controls the actual production of the movements. And what I argue is that during embryonic development, this motor learning pathway is replicating multiple times to control the hands, to control the legs, control the chest, and so forth. But it's missing in the vocal learners to control the voice, the non-learners, that is. I argue that in the vocal learning species, in humans and parrots and songbirds, it replicates one more time through some kind of genetic mutation, and this extra copy of this motor learning pathway takes over the pathway that controls vocalizations, and this extra copy then becomes a vocal learning pathway. We call this brain evolution by brain pathway duplication. Like gene duplication. Has anybody heard of gene duplication? Where you get a copy of a gene, copies itself, and then one goes on to have a new function, the other keeps an old function. Well, the old function is learning how to walk. The new function is learning how to talk. All right? And so after that happens, then we think what happens is that not only does the areas, well, you have to have auditory information from the ear get into the vocal pathway to control the voice. And what we think happened, or we're proposing happened, is that once the auditory signals went inside of this vocal learning system here, you needed tight integration between the sounds you hear and the sounds you produce. The larynx is the fastest firing muscles in the body. You need to integrate that, and there are some specialized mechanism for making that happen, we think, in the vocal learners, contaminated the surrounding motor pathways, allowing us to learn how to dance. All right? And now it's bringing me back home to my first passion. So for the third part of my talk, we're going to test this hypothesis. Because if this is true, it should mean that the underlying genetics of the pathways that control learned movement should be similar to the pathways that are controlling the vocal organs, even including the brain pathways that set up, the genes, that is, that set up the brain pathways. So to get at this, what we did is, like you see on CSI television shows, we use lasers underneath a microscope to dissect out with very sharp precision the brain areas involved in learned communication from these vocal learning species. And we put them in little test tubes and then isolate DNA and protein and so forth from these cells that are inside the vocal learning nuclei versus the outside. And we do that for each of the vocal learning species and their closest relatives that don't have the ability to imitate sounds. And we have several tools or ways of then measuring all the genes, not just that one gene I showed you that was responsive to activity, but almost the entire genome. At the first, we use what was called microarrays, where you have glass slides, where each dot here represents one gene. Now we have new techniques where you can actually sequence all the messenger RNA in a cell and say who's there and who's not there. And we did that for the birds, and the Allen Brain Institute, some collaborators of ours, were doing it for humans and non-human primates. They got 3,700 samples from 6 postmortem brains, people who donated their organs to science, after they died, of course, and several hundred samples, yes, and several hundred samples from 6 monkeys, and also then interrogated to see what kind of genes are present in many, 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 many different brain areas of humans and non-human primates. And students and postdocs in my lab who all now have their own labs, also of diverse backgrounds, then developed the computer science tools to analyze thousands of genes, gene products, messenger RNAs we're seeing in the cells of birds and human brains. And what we found is that the messenger RNA products were different for many genes inside versus outside the song learning nuclei, or inside versus outside the speech areas of humans, such that those differences were shared between some vocal learning brain regions in songbirds with the laryngeal motor cortex for speech in humans, or the basal ganglia region for learning how to imitate song in birds or speech in humans shared some specializations. And more recent work from Gregory Jedman in my lab, who's here today, and you can talk to him during lunch. He used some novel new technology that looks at all the genes and found that this region, HVC we call it, the high vocal center, is also similar to another cell type in the human laryngeal motor cortex. And so when we look at these specializations, genes that are turned off or on relative to the surrounding brain area, and ask, do we see them in a chicken or a quail brain or some other non-vocal learner? Do we see it in a non-human primate or even a mouse? And the answer is no. So just like the circuit itself is convergent, so are some of the genetic underpinnings of the messenger RNA products are also convergent. And what do I mean? What does that look like? Here shown in the actual brain section, remember this white signal is the mRNA. For this one gene called SLIT1, it's turned off in each of the vocal learning brain regions of the vocal learning birds for the ones that produce the sounds. Here in a dove, it's basically a pigeon, or a quail. What's the closest relative of quail that somebody might know of? I heard it, starts with a C. Chicken, there you go. All right. That these species do not have this downregulation. Here in the human cortex, this is the strip of cortex that controls movement behavior. It's downregulated here in the white signal. And other colleagues of mine who actually are neurosurgeons who go into the human brain in patients who have tumors or epilepsy, and to avoid speech areas, they put electrodes on top or into the brain to record different areas and ask the person to speak. They're awake during the surgery, of course, locally anesthetized, and ask them to speak, and then they see, okay, where are the brain areas that show the highest activation in red here doing speech production, and it's exactly these regions that show convergent gene expression with the songbirds and parrots and hummingbirds. So that's very interesting. Other work happening in my lab, Lindsay Cantin, who's also here today, a graduate student, is trying to figure out why are these genes specialized? What's causing them to do that? And we're finding that remember I told you the messenger RNA? Everybody heard about that, right? Has anybody heard of the coding sequence? That's what this is. This is it's called NeuroD6 gene. Neuro, it's expressed in the brain. It regulates other genes. The messenger RNA for this gene is regulated by a regulator that binds to what? What would this be called? Like a promoter. There you go. And so we're finding that there's certain genetic changes that allowing regulatory enzymes to bind to the promoter inside the vocal learning area, but something is blocking it, some proteins preventing it from binding outside, which we then think causes regulation of other genes, not just one or two, but many other genes. And so, what are causing these specializations is that could be one reason, but what are the specializations causing themselves? Why turn off a gene? Okay? Well, it turns out this gene, SLIT1, that I told you about before, here, let's say if this were in the cortex, this is down here in the brainstem controlling the voice, its receptors express at high levels in the neurons that synapse on the muscles, all right? When SLIT binds to its receptor, it does something that's kind of counterintuitive. It says, please don't connect me together. Please avoid me. So when these neurons are sending, who heard of axons? You heard of axons, right. When the cell bodies are sending axons down to the brainstem that control the muscles, they're saying, avoid me, don't connect to me, okay? And connect to some neurons around me. But we think in humans, and songbirds and parrots, when the SLIT1 is turned down or turned off, it says, okay, the ones that have high levels, it says don't connect to the neurons that control the voice, but the ones that have low levels of SLIT1, it says, okay, you can connect to me, you can talk to me directly, and then control the voice directly from the forebrain. So how does that look like in a human brain? Here is the part of the human brain that controls the ability to produce speech, and it makes this direct connection to the neurons that control the voice, whereas in a non-human primate, it makes an indirect connection or no connection at all. So that's simple. I don't think that's the only difference, but that is one big difference we're seeing consistent across species. And what does that mean? Well, in science, we like to make hypotheses, but we have to do them? What we have to do with those hypotheses? We have to test them. Well, how do you want to test this? Well, one way of testing is we can turn on the SLIT gene here in the songbird nucleus that controls vocalizations and try to make a direct projection indirect. Or we can turn it off here in a chicken, right? And maybe take over an existing motor learning system to make an indirect projection indirect projection direct, and do the same thing in mice, but we actually have no idea what to do in mice. And because no one actually tested their systems for vocal behavior. But imagine that, if we can actually make a vocal learning system, have a connection like humans in these chickens or mice, what do you think they could do? Right. So when we realized that, we thought, oh, this is a really cool experiment. We actually convinced some people to give us some money to test that idea. All right? And that's the last part of my talk. To not ruin the story, that we haven't gotten there yet, but we're moving along. And I'm going to tell you, this is, if students want to join the lab at some point or come, this is one of the areas of research we're actively pursuing. So, but it turns out many people assume that mice don't produce learned sounds until it was found by Tim Hawley and others. Here's a sonogram of the sound. Mouse vocalizations in the ultrasonic range, pitched down by computers to our hearing range, sound like this. That sounds like a what? It sounds like a bird, right? Yes. And we see all these different kind of syllable types. This is time, and this is the frequency of the sounds. And mostly the males produce these long sequences of sounds. And when they do, they chase around females, all right, to try to mate with her. So these are what we're calling love songs. And if the songs have these more complex syllable types to them, the females are more receptive to them. If they're singing the boring, simple ones, the females say, get away from me, all right? And it was found that different brothers, these are three different mice here, right? And these are three different syllable types, that different brothers had different proportions of syllable types that they produce, indicating it may not be totally innate. And so this prompted a number of labs to investigate this, and a number of labs said no vocalizations at all. Yes, mice are just like everybody else. They're not like humans, right? And others said, oh, maybe a small amount of vocal plasticity, ability to modify their sounds, but still nothing like what we're seeing in songbirds and humans, even though it sounds like a songbird. Some work from postdocs and graduate students in my lab. What about in the brain? We found that there is an area of the forebrain here in the mouse, the cortex, that shows similar gene activity when they vocalize. That was a surprise because it's thought to be only in humans. And when we started looking at the connectivity of this region here by injecting what we call neural tracers in the muscles that go back up into the brain, we found that there are neurons there, now let's blow that up, you can see these individual cell bodies and their dendrites here that send the projection directly down to those neurons. So if we put another tracer here, we can see the axons. Can people see these little black squiggly lines back there? Maybe a little bit. The brown cell body here is the cell body that controls the larynx. And it's receiving 1 or 2 direct connections from these neurons here, but not hundreds. We see hundreds in humans and song-learning birds. So if we look at the SLIT1 expression in these mouse brains, here again is a reminder of what we see in the vocal learning birds and humans. The gene is turned off. In the mouse brain, it's not. Okay? So what this leads us to is what I'm calling a continuum hypothesis. The world is not just black and white. All right? Neither is something like spoken language. What we think could be going on here with vocal learning is that there are very limited or no vocal learners, like some lizards that can't even vocalize at all. Okay? There are some others, like mice and maybe some non-human primates, that can modify their vocalizations to a certain degree. Then there are the complex vocal learners, like songbirds, that just learn how to imitate their own sounds. And then some parrots and other birds that can actually imitate other species, like humans. We're calling them the high vocal learners, with humans being the most advanced. So we're calling this a continuum hypothesis and a step-like function here. And mice are somewhere here with a small number of connections from the forebrain, but not like what we're seeing in songbirds or parrots and humans. But with those small number of connections, maybe testing a hypothesis can be made easier. Instead of trying to genetically induce a vocal learning pathway, maybe we can take an existing, very minor, tiny connection and genetically enhance it, okay? And so that's what we've been trying. We've been, this in red signal is the SLIT1 protein, and we've been able to upregulate it in a vocal learning species, the zebrafinch. And what happens, the more higher expression we have of SLIT1, the more we turn on the gene, the more we see deterioration of the learned sounds of the bird, of a song-learning bird. What about mice? We have been able to turn it off, hooked up to green, what's this? Some of you might have heard about this. GFP, what does that stand for? Yeah, green fluorescent protein, right. So here we have an inhibitor of this gene hooked up to a green fluorescent protein so we can see that it's there. And these mice, what they did is they just didn't like to vocalize as much, which was disappointing for us because we wanted them to vocalize more. But work, again, from former students and postdocs in my lab. A recent postdoc has, however, been able to get mice and genetically enhance them to knock down SLIT1 in the genome itself, such that it's not expressed in any of the neurons that project to the forebrain, the brainstem. And what Lomax has found is that these mice produce what we call homozygous knockout. What is a homozygous knockout? Yes, where both copies of the gene are actually mutated. And these mice actually, with less SLIT1 of both copies, or actually none, are producing more complex syllable types than the mice that actually are heterozygous, one mutation or no mutation at all, which is consistent with what we think could be happening. The less of this gene, the more complex the behavior could be. So this particular gene, SLIT1, is actually regulated by another gene that's even more famous called FOXP2, all right? FOXP2 binds to the promoter of SLIT1 and turns it on or off. And what's interesting about this gene called FOXP2 is that mutations have been found in humans, particularly first in what's called the KE family, that children who have these mutations have deficits producing learned speech. And here's an example.
Your name?
Laura.
She's trying to say Laura.
Where do you live, Laura?
She's trying to say Sheffield.
And how old are you?
4.
She said 4. So Laura is a 4-year-old child, has very good auditory learning, can ride a bike, can walk to school just fine, can eat just fine, but has difficulty producing learned speech, all right? And so we asked, what if mice have the mutation that Laura has? And so working with Simon Fisher, we put this same mutation in the mouse genome, the one that Laura has, that inactivates one copy of this gene. And what we find is as follows. So the black bars are wild-type mice. When we put a live female in the presence of a male, the males go crazy producing these more complex syllable types that the females like, that I told you about earlier, right? We call them the sexy songs. Unfortunately, the mice with the FOXP2 mutation, yes, they can sing, but they don't switch to these more complex syllable types as readily as the wild-type mice do. And so what about the neurons that control the vocalizations, the vocal organ itself in the forebrain, or that at least connect to it? We see them there in the mice with the FOXP2 mutation in the cortex, but the distribution in the brain compared to wild-type, they become more spread out in the cortex instead of being localized. So this FOXP2 is necessary to localize these neurons that then connect to the organs, or the neurons that control the organs for vocalizations. Well, I'm going to wrap up now. I'm going to say that that's where we are so far. There is interesting special issue of Science magazine recently published, including an article that I wrote that has a lot of the things that I talked about, if you want to look at it further. I try to make it more digestible for a general audience. So since you answered my questions very well about messenger RNA and all the other things, you should be able to understand this and some of these other articles. I encourage you to go look at it. And to keep in mind two things when you walk away. The motor theory of vocal learning origin, that spoken language is really a movement behavior. All right? As complex as we like to think of it, it's not as, it may not be as high in the sky, you know, whoa, whoa kind of behavior. It's actually something that's similar to like walking and flying. And that all of us, it's not a black or white world. A lot of biology is a continuum. And to think about that continuum and what happened over the last 300 million years, here is the cortical regions of all the different vertebrate groups. And in the vocal learners that we've seen so far, humans and these birds evolved some similar brain circuits that control the vocal behavior itself. And I predict if a lizard or some crocodile or some frog were to evolve vocal learning another 500,000 or 1 million years from now, I predict that you're going to find these brain regions, because evolution likes to repeat itself. So we're getting closer to understanding the reason why these close relatives can't do what we're doing, and how language is related to other kinds of behaviors. And we're doing that also by, one thing we're going to do is we're embarked upon a project not just to look at the genomes of species that I just showed you, but to get the genomes of all vertebrate species on the planet, all 66,000 species. And we don't have the money for it, but we have the ambition. And so, and this has become even more important, not just for understanding, are there any other vocal learners lurking out there that will see signatures in their genome of these genes like SLIT1 or FOXP2? And we don't know, but I'm going to predict there are. But another reason that this is an urgent thing for us is that, like the fires in Australia and what's going on around the planet, species are starting to go or getting to the brink of extinction much faster than ever before, in what we're now calling a sixth mass extinction. And so we're also in this project trying to capture, whether they be vocal learners or not, the genomes of species that are about to go extinct, so we save their genetic material, hopefully forever, and maybe can resurrect them one day. So I'm going to end, the numbers are incorrect there, but I'm going to end here in 3 points. Complex behaviors can evolve multiple times from deeply homologous structures in the brain, like motor learning circuits. A trait like spoken language can be understood by studying other animals, and convergent evolution of mechanisms of how a behavior works also is associated with convergent mechanisms of the disorders of that behavior, like even for speech. So yes, we can study songbirds for speech disorders. All right, here is my lab. A lot of people involved in these various projects. I gave credit for those that I had shown throughout the talk, and some funding sources. And I will take your questions now. And by the way, we have ticket awards for those who ask questions. You get 2 free tickets to the American Museum of Natural History. Thank you very much. Yes. Okay, I'll give her the microphone. Go ahead.
I have a big mouth already. I'm their teacher. They'll tell you that. Do you find that there is a difference with regards to the downbeat dancing versus swaying, for example? Because I have one African gray that likes to dance, and she does downbeat and she does sway. And my son Kanye, who prefers classical music, does not bob or downbeat, but he prefers to sway instead. So what could those differences in how they reflect their preferences be indicative of what you're talking about?
Yeah, just like humans have different personalities, well, so do non-human animals, including for dance. So a paper came out recently showing that Snowball, and they think other parrots, over time actually refined their dance movements like dancers do, and practice. And they then add or take away to their repertoire. And different birds will, some like when you're saying swaying back and forth versus bobbing their head. So I'm just going to, without much more evidence, say, based upon what we know so far, these are just personality differences. But they're both dancing. Yes. Yes. And you get a free tickets to the American Museum. Yes. Is the question back here somewhere? Jeannie, could you help me get it out? Yeah. Sure.
Hi, I'm also a teacher. I've been interested in the idea of neuroplasticity, and in terms of teachers and students, we often think about practice helps to improve brain connections. Mm-hmm. And so I was curious for your research, as you're starting to look at these brains and perhaps remove inhibitors of speech and get animals closer to that threshold, do people ever look into how can you encourage animals to practice skills to encourage the growth of these brain areas to get them to do what you want to do?
Yeah, what is it? The stick and the carrot approach. Even in science, scientists are using sticks and carrots to try to train animals, and some believe, I hope the evidence is more in favor, give a carrot, the reward, that is, and instead of punishment. But practice and brain growth. Actually, work that I did here when I was a postdoc and followed up on by some others is that we found that when songbirds produce their learned sounds, when they sing a lot, all right, that singing, many people don't realize, but these birds, they actually have new neurons that are entering the brain even throughout adulthood. And what we found is that when they're singing a lot, the birds who sing more have more neurons that are surviving and staying healthy in their vocal learning brain regions than the birds who are quiet, all right, for a long time. And so this basically called as motor-driven neuronal survival. So you should think of the brain like even the parts controlling my hands here, or the parts controlling thinking, the parts controlling the voice, that it's not quite use it or lose it, but is use it and it keeps it healthy, just like exercising in the gym. So basically, thinking thoughts for the visual cortex, hearing yourself, imagining things, is causing those brain pathways to stay healthy. So please use your brain, all right? Just like you're in a gym, all right? Yes. Okay, over here? Okay, all right, up here?
So what's the difference between the comprehension that the parrot is having versus the gorilla, and how that works with speech, because speech also was part of, like, the difference between the comprehension that the parrot has versus the gorilla, and how it's being replicated through.
Yeah, this is a good question in that, is there a difference between our ability to understand speech or learn sounds and species that don't have the ability to produce them? That is, do we understand, learn communication or learn speech better than gorillas? Coco understood about 7,000 words. She unfortunately died a few years ago, but before she died, after being with humans for 40-something years, she understood about 7,000 words. Our everyday vocabulary is about 20,000 words. So let's say less than half of a general vocabulary of human speech sounds you can understand. That's quite remarkable. Can a mouse understand 7,000 words? I don't know. I don't think so. But I think there are going to be differences in species in auditory comprehension, but I don't think it's going to be quite correlated with the ability to produce sound, probably more correlated with other things that animals have to do. Do they use hearing versus vision versus smell? Mice like the smell. Mice actually can interpret a lot more different smells than we can. We're kind of bad at smelling compared to mice, but we're better at auditory comprehension. Does that help? Yes. Okay. Yes. I will let you folks control who gets the answer. I mean, the question over here in the corner. Yes.
Hi. I'm not sure if this really pertains to the discussion, but I remember years ago I had an accident and I was in the hospital, and for whatever reason, for a certain time frame, I couldn't speak. So I had to write down information.
Mm-hmm.
So I'm trying to figure out if trauma triggers the gene to be turned on so that it prevents you from speaking. I don't know what the exact question would be.
I would say trauma turns to turn on the genes that are involved in brain repair, and including in humans, you can actually induce new neuron formation after the brain injury. Normally, human brain doesn't produce new neurons, but in brain areas of injury, this pathway will turn on. And people are trying to study how to make it even better so that you can actually really repair the brain. But in your particular situation, I'm going to guess that the accident that you experienced impacted the speech production, the vocal production areas more than they did the areas that are involved in perception. So this is why you could do what you were doing and understand and kind of know what you want to say, but have a more difficult time saying it. This happens often as we get older. It's called aphasias. I'm not saying that's what happened to you in particular, but where some type of brain damage or degeneration or some kind of trauma, a stroke will prevent not only movement of the arms and so forth, but an inability to move the larynx, or one in the back of the brain here, the larynx is okay. You can say lots of different words, but in your perceptual area, you're having difficulty actually understanding what they mean. Yes. And that's the separation between vocal learning and vocal production. Okay, you had your hand, okay.
Oh.
Oh, okay,
Yeah,
we'll
sure,
come to
go
you
ahead.
next, all right? All right. Yes. If we can give him a microphone. All right.
Right
Back here?
Sorry, so along those lines, I was wondering whether, I know a lot of students or children younger get speech therapy. So I was wondering if it's something in their speech learning, or their genes are being turned off or turned on, or how exactly going to speech therapy affects their way of producing the speech.
Yeah, that's interesting. In speech therapy, including speech therapy in autistic children, there are different kinds of autistic traits, or let's say behaviors. And one class is where children, again, have, like in aphasia, have decent ability to understand what someone is asking them, but difficulty producing speech. And what's thought to happen there, and this is what we call a hand-waving hypothesis, because it's not really demonstrated, since the brain pathways for speech are thought to evolve later, after the brain pathways involved in comprehension, that what's happened is that they can be separate in terms of their development. And that in some autistic children, there's a deficit or a speeding up of the critical period that it didn't give time for the brain pathway, the more later evolved system, time to catch up for the other critical period behaviors. And so it gets halted, basically, in its development. And then you need speech therapy to try to work those brain pathways to finish developing and be able to produce speech, even though the comprehension is okay. I kind of muddled that, but I think you got the point. Okay, you can pass the microphone down here. Okay, good. Yes.
Hi, so I was wondering how exactly these genes react in people who are born deaf or mute. What exactly kind of stops that auditory learning? And
Yeah, so we've looked at this actually in the birds, and some people have looked at not necessarily the genes, but the brain regions in humans, what happens to them in deaf and mute. In the birds, we found that if you mute the bird, basically the mute means that you can't control the muscles from the forebrain. If you do that, the bird tries to sing, but it can't come out with the song. And what we see is the genes in the brain turning on, okay, they're active. They're doing what they're doing, but the muscles just can't get the signal. All right, and so that's what's happening there. In humans, it was found that for deafening, when you become deaf as a child, before puberty, basically, before the brain sets in with your hormones and so forth, then what happens is that the brain areas that control the auditory processing, hearing of sounds, get invaded by the brain regions controlling vision. So in other words, your visual regions become bigger. And that enhances your visual ability in deaf people who are deaf at an early age. And we've seen something similar for birds in terms of the gene activation, something we just haven't published yet, but we've seen it there as well. So we think the deficits, as I said in my last slide, that are occurring like deafness and muteness are similar in birds and in humans. Over here.
So people with the FOXP2 gene, like Laura, are they ever able to overcome their speech impediment or manage it better, or will they have like a disability to speak properly for the rest of their life?
So far, no, unfortunately. With some speech therapy, yes, you can overcome. So Laura has one copy that functions fine and another copy of the gene that doesn't. And so, so it's not like they're totally completely impacted. So for some therapy and practice of speech, they can get better, but they never get to the point of a normal person. We hope one day this would be possible. So these mice, we have the mutation in them. We can now test gene therapy tools on these mice to see if we can inject something in part of their brain or give them some drug that enhances FOXP2 activity and see if these mice can overcome the deficits that they have and whether those females will like their sexy songs afterwards. Yes? Yes? Oh, over here? Okay.
Are parrots able to produce sound? Are parrots able to produce sound that's not imitated by other like humans, like on their own?
Yeah, so what's interesting is that all vocal learners, including us humans, like we cry. Cry is an innate sound. Most of the sound of crying is innate, or ow, or something like this. So parrots, like us, also produce innate and learned sounds. And that's one thing to know. And then the second part of the answer to your question is that parrots are not just imitating humans when they're around humans. They can imitate car alarms, they can imitate phone rings, and why do they do this? Well, in captivity, one reason is thought they're trying to get your attention. So they notice that you start talking to somebody on the phone when your phone rings and so forth. So they want you to talk to them. All right, so they ring the phone so you can start talking to them. All right, and so one of the motivations for humans and in parrots is that we naturally learn speech. We don't just, yes, you're in the classroom, you have a teacher up there on the board or some presentation trying to teach you didactically, but you're going to learn speech even if you didn't go to the classroom. Doesn't mean you shouldn't. And so why is that the case? Somehow the mechanism to learn speech, to learn parrot vocalizations, is built into a socialization mechanism. We learn speech because it's rewarding. Dopamine in the brain is being released as we're learning how to imitate sounds, or the parrot is ringing the phone and getting a reward by the person saying, "Who's that? Who's that?" In the wild, though, parrots will imitate sounds of other species to increase their repertoire. All right? The bigger their repertoire, the more attractive they are to the opposite sex, so the more intelligent they seem. And so there you go. Yes. Okay, over here, we'll flip back and forth. All right. Yes.
Hi, so recently I watched a BBC documentary on the planet of birds, and it showed one of the songbirds showing their repertoire to a potential mating partner, and it was sounds like from a distant village, wood getting chopped, children playing, and it was a lot of variations of voice tones. And I was just wondering what makes a songbird have that high of an imitation skill level compared to humans.
Right, so I haven't seen this document. So they were saying the songbirds were actually producing sounds in various different contexts.
Yeah, it was just different things that it's imitating from like nearby villages to impress?
Yeah, so I think in this case, when it is imitating many different sounds in the environment, it is trying to increase its repertoire. There are some studies that show that the birds are not just some species of birds, like the chickadees and so forth, or starlings, are not just imitating sounds to sound sexier, or more intelligent, but are actually using their imitated sounds to have meaning to others, to communicate what we call semantic information to others in the community, like a predator is around. And they have certain calls that indicate the size of the predator and what kind of predator. So this has led to a theory that in most species, we're producing learned sounds for attraction or territorial defense and so forth, including humans like Jennifer Lopez, I imagine. But in some species, like what you're describing here and what I'm describing, and in some parrots, they also produce imitated sounds to communicate information about the environment around them. Oh, we got lots of questions here. We're going to run out of tickets. All right. Yes.
Hi, good morning. So I was just wondering, so in addition to being a student, I'm also a singer, and I was wondering if there was some sort of similar pathway that's activated in the brain when singing as opposed to speaking in people. And in addition, just as an extension, I was wondering if the genre of music has some sort of effect on this pathway being activated.
Yes, I'm going to say, well, I'll say what's known, and then I was saying what could be hypothesized, is that it is the case that not necessarily so much singers, but folks who are musicians tend to do better in math and language tests. And it's not thought to be something genetic, but something thought to practicing music. Music is like a language itself, right? For singing ability, and this is one of the things, a lot of my family sings, and they're great singers. Some are even professional singers. I wasn't as good as them, and I've been wondering, what do they have that they don't have? So in the future, I'd like to get your genome and others, right, to find out, is there something genetically different between the good singers and the not so good singers? And is there a relationship there between dance or not as well? We don't know the answer, but there is a suspicion that there is some genetic difference in people who have a really good ability to sing and those who don't. And what that genetic difference is, we don't know. And by the way, for the ones that are like me, just because you may not sing as well as your relatives doesn't mean I'm the only one that became a scientist, so the two aren't correlated. All right. Yes. We have some more of these tickets down here if you want to give them out as they're asking questions. Sure. Oh, okay, go ahead.
You mentioned earlier that in people who are born deaf or mute, their vision takes over. And I was wondering if it goes the other way in terms of if you can put a dampening on vision in mice, then their caliber for exercising vocal production would increase as well.
Right. If a mouse goes deaf or a bird goes deaf, then what?
The visual.
Ah, yes. Okay. Yeah. So, I can't recall of studies. I can imagine this would happen, but I can't recall of studies that have really tested that. That is, if you lose vision, right? Does your auditory cortex now take over your visual cortex? But there are some studies in kinesiology. Has anybody heard of this? Synesthesia, there you go, synesthesia, where if you see something, you hear it, okay, and vice versa. And one hypothesis behind this is that there is the auditory cortex, visual cortex overlap, or is occurring too much in that person who is having this synesthesia effect. And so this would be some evidence that could happen. Wow, we're getting more and more questions. Yes. Okay, this side, I'm letting you folks pick out who's. Does somebody have a microphone on this side?
Right here.
Okay, go ahead, yes.
So I wanted to know how the brain development of a human would be affected if they didn't grow up around humans, like at all.
If the brain, say it again?
How the brain, like a human brain would be developed if they didn't grow up around humans at all.
Ah, like feral children. Okay, yes. And this has happened, there's some famous cases in Europe where children have been left out in the woods and raised by foxes and so forth. and it's been done experimentally in birds and other species. And so what happens in the birds is that if you take a song-learning bird and remove it from its conspecifics, raise it alone, or raise it with some other species that's different from its own, but particularly raise it alone, it produces what we call isolate song. It doesn't make sense. It's all varied and so forth. It doesn't crystallize, but it's also not species specific. You take another bird and raise it alone, it will produce a different set of strange kind of sounds. Well, this is what seems to happen with feral children, is that if they're out there in the world, they don't have something they can hear to imitate. So they produce some kind of novel sounds that sound something like humans, but aren't quite any language. Now, if that bird is kept in isolation past its critical period, past the puberty-like phase of life, and grows up to become an adult, and then you try to teach it new learned songs of its own species later in life, it has difficulty doing that, all right? So you learn how to imitate sounds best in your critical period, and it's difficult later on. And there are some species of birds that where after adulthood, they can't learn anything new, so forget it. And there are other species like humans that can continue to learn something new. And this is thought to be similar with these feral children. Fortunately, there are not enough cases out there to do statistics and say this is really rigorous science, but the anecdotal evidence indicates it's similar to these vocal learning birds. On this side?
So those with disabilities and unable to communicate with others efficiently, you were talking about the correlation with other animals, and I wanted to know about they're missing certain areas in their brain production, and how does speech therapy kind of influence that?
Yeah, so they're either missing it in some species, or it's there, like we see in these mice, in a very rudimentary form. And so in those species that are missing it or in very rudimentary form, there's no amount of therapy you can give to them that's going to get them to imitate sounds. For those that have the circuit, but it's not properly connected, or it is something genetically modified about it, therapy can help them. And so how that might differ from one person to another, we don't really have the answer yet for that. We need some students to actually join our research and figure it out. Yes. Okay.
Would a parrot only have to hear something once to mimic it? And also you mentioned that parrots can do things like crying and stuff like that, can mimic noises like crying and things like that. Would they also be able to mimic things like yelling or whispering?
Yes, they can yell, they can whisper, and there's some species that have one trial of vocal learning. Not all of them. There are 4,000 species of songbirds, by the way, 300 of parrots. And they have different vocal learning skills to various degrees. So it depends. An African gray parrot does have an ability to imitate on one trial learning. Not as good as humans. Humans are the most advanced, but still decent compared to other birds. Whereas a small parakeet, a budgerigar, needs to practice over and over and over again to get the imitated sound correct. Is that it?
I was wondering about how you were talking about how there's different type of neural pathways for auditory things and physical
where
things.
Where is the person at? Oh, here. Okay, good.
Yeah, so I was wondering, for people that are deaf and they have to use sign language, does that use a physical motor pathway or an auditory pathway?
Very good question. So, and there's a debate about this in the field. I'm going to tell you which side of that debate I believe. Is that when we do sign language, the brain pathways that control the signing ability are adjacent to the brain pathways that control the ability to speak. But when we perceive sign language, right, if you're a deaf person, you perceive sign language only in your visual pathway, or the part of your visual pathway that is taken over auditory, right? But if you're a hearing person and you associate signing with sound, your pathway for vision and your pathway for hearing, auditory pathway, are both processing those sounds. And further, what's thought to happen is that, make a sign gesture now. Let me see you. Some sign. High, he's high, okay. And so that stimulus that he did like that went to my eyes. From my eyes, it went to my visual cortex. From my visual cortex, it went to my speech cortex. And the speech cortex said hi, okay? And the speech cortex said hi, and my auditory cortex heard me say it inside my brain, all right? And this is one hypothesis of what's happening, and also how we understand or how we translate one type of stimulus in one sensory domain to another. Yes?
Okay. Like, this might be a really stupid question, but do you think...
no question is stupid in this audience. Yeah.
Okay, do you think that epigenetics could have something to do with auditory comprehension? Like, for example, like what she mentioned with like her trauma, like do you think like some type of methyl groups or like histones could turn off certain genes in the brain, cause like a speech impediment, and that could be passed on to like future generations?
Wow, I see your teachers have been teaching you well. Okay. Thank you for that question, yes. So epigenetics, this is a modification of the DNA through proteins or other molecules. And one of the slides that I showed you that Lindsay Kenton, who's here, work is looking at the epigenetic modifications of the brain cells that control vocalizations versus the adjacent ones that control other behaviors. And we are seeing differences. And so, so now you're asking also about not just the vocal pathway, but the auditory pathway. One hypothesis is that epigenetic changes are occurring on the genes in the auditory system or other brain regions as a result of learning to help stabilize memories. And so it could be possible that, yes, there are epigenetic differences as a result of auditory learning, but no one has showed that. So I'm just saying it's possible. But what Lindsay has found is that there are epigenetic differences for the vocal behavior versus non-vocal behaviors. Yes?
So I just have a question about, I know you said that the size and this in the brain of the bird versus how the human has more surface area. I know you said that at first it didn't affect it. But with the combination of having those areas in the brain where you could vocally talk, would that increase the ability of the bird or the species to be able to talk, like the bird disco? Do you think that would have anything to do with it?
Yeah, we think there are multiple variables that are increasing the ability to produce, learn complex speech or to sing better and so forth. And I don't want to say size doesn't matter at all. I think the size of the brain region matters to a certain degree, but not as much. Two other things that will matter, I think, is genes involved in what we call synaptic plasticity. That is, the ability to form and break connections in the brain more easily. And those genes, if they're turned on at high levels, we think it allows us or animals to learn faster, better, and more. But if you keep it too plastic, all right, throughout your entire life, it means you also forget a lot. So you got to have a balance, all right? All right, the most exciting thing, though, that seems to make a difference, or we think is correlated with a difference, is that it's recently discovered that all of the song-learning bird species have not necessarily bigger brains, but more neurons, more tightly packed in a similar space. So songbirds have twice the number of neurons as all other birds. And parrots and crows, which are also songbirds, even have more neurons than that, such that a parrot has the same amount of neurons as a monkey 5 times its size, all packed together. And so we think when the extra brain pathways evolve for vocal behavior, other brain pathways didn't disappear. It just says, move over, give me some more room, and pack them tighter and tighter together and make all these connections that then control the vocal organ. Yes?
I'm sorry, but if humans are the most advanced, like, what do you call that? Species, how is it that these less advanced animals can understand our language, but we can't understand theirs?
That's a good question. Very good question, yes. She should get applause for that one, yes. Right, I think we humans sometimes, despite all of the achievements, we sometimes overrate ourselves, okay? And yeah, so we are having a very difficult time understanding what are these animals saying to each other. We get some hints at it, and just to think about it, everybody here is understanding what I'm saying. I'm sure, I believe they are, at least understanding the individual words, if not the actual ideas. But if I were to speak in some other language, particularly, some people are going to send this, arigato gozaimashita, right? Right? You're going to say, what did he say, right? Does somebody know what I said? Thank you, that's right, yes. Okay, too many people know Japanese. So when I hear some foreign language that I've never heard before, I'm like, what in the world are they saying? And I can ask them what they're saying. We can talk to each other. It's hard to communicate with an animal in that way. So I think once we get the ability to genetically modify these animals to get them to imitate more, we'll start to also be able to get them to imitate sounds in a way that we can understand them more or talk to them better. Yes.
Hi, so you talked a little bit about how your findings have impacted neurosurgery techniques. I was wondering if you could speak about how understanding these connections, understanding the genes can improve that, and kind of building off of that, can gene therapy be a pathway for treating some speech disorders, for example?
Yes, I'm hoping that it can be. And as we modify these genes and manipulate them in mice, we'll be able to test those ideas. Gene therapy is being done on humans, either by local injections somewhere in the body or into the bloodstream. But it's a really dangerous and tricky approach because you don't want to have unintended consequences where you modify a gene to, like with any drug, to enhance a brain circuit, to repair it, cause the neurons to form the right connections, but then it does something else different that you didn't know because there are almost 20,000 genes in the genome, and by manipulating one gene and manipulating some connections, you don't know how you're going to affect the others. So we need to do a lot of work to make sure that it's going to work well. And a mouse is not a human either. So there might be some effects that occur in humans that doesn't occur in mice, and you won't know until you test it in humans. And what was the first part of your question?
How this can impact neurosurgery techniques.
Okay, all right. Neurosurgery is so far advanced. I'm amazed, with all these tools. Some physicians, they can control these instruments without touching the patient at all with all these different dissection tools. It's really amazing. But for this to work, it can be done. It actually can be done on humans. And in some countries, they are trying, where you inject locally in the brain in some brain regions some viral molecules that have been modified not to harm an individual, but to get inside the genome of the cells that it's injected into and affect behavior. So I think it's probably going to happen in your lifetime. I don't know if it's going to become a standard tool to do this during surgery, but I think it's possible because we do it to animals right now. Yes, way back here? Go ahead.
Hello. So, in my opinion, we study some human-made language, which like English, others. It's because we have a lot of information need to talk about, and we need to use them for a better future like that. We need to create something. So, if like animal, they need to do something to save their life, will they increase this hobby and like some ability?
Okay, yes. So it is the case that we humans, compared to all other species, except the ones we domesticate, are sheltered. So we don't have to worry about being eaten all the time, or where we're going to find our next meal, as long as you're getting paid well, hopefully, right? Or even if not, And so, so we've gone on to use our learned sounds in things like I'm doing now, which is to communicate complex ideas and not worried about some wolf going to come and eat you while I'm talking to you now. But with that said, the vocal learning species out there in the wild, including parrots and in elephants, they do communicate with each other socially. they do form complex social networks, and they do have some free time to play. So it's not all, dog eat dog out there in the wild. There is a level of social component from which we inherited that ability and just took off with it and doing more with it. What's interesting about that, I'll say, is that all vocal learners are either at the top of the food chain, like humans or elephants and dolphins and whales, or they're producing ultrasonic learned sounds like bats in a range where others can't hear them. And what I think happened there is that all these learned sounds producing are highly varied. And if you're out there in the wild talking a lot and talking all these different sounds, predators, their auditory system can't habituate that easily to your talking, right? And we vocal learners like to talk a lot, right? And so I think you're more likely to be eaten unless you can avoid predators, and this is why vocal learning is so rare. Yeah.
Hi, so I wanted to know, certain diseases that migrate to the brain end up slurring the speech or having you confuse words. If it worsens, could it possibly stop the signal that allows you to speak? And once you are cured, does that, because it did the signal was interrupted, does it eventually come back again with therapy, or is it like just turned off?
Yeah, so in some cases, a stroke to the frontal parts of the brain that control speech. In the initial stroke, persons are more paralyzed and have more speech deficits than, let's say, 2, 3, or 4 months later. So there is some recovery. So there's the initial shock followed by recovery. And why that's the case, there are two reasons thought to be the case behind it. One is, it's just like you fall off a bike. You have that initial pain, right? And you're in excruciating pain. But eventually it goes away, and then there's a numbing feeling that lasts for weeks afterwards. It's the same thing in the brain. There's the initial shock and trauma where the neurons aren't firing, and it's just saying, pay attention to what happened and try to figure out what's going on. I'm doing a euphemistic thing. I'm not sure that's what your brain is doing. All right. And then there's recovery. And that recovery can involve some new neurons, as I was saying earlier, or even old neurons that are just getting replenished with fresh blood flow. Yes. Okay. All right, last question. Go ahead.
Can you hear me? I actually had two questions.
Okay, last two questions.
Children who has weakness in auditory sequencing.
Say it again.
Auditory sequencing usually have problem recalling the order of words.
Yep.
I was just wondering, how does that happen, and is that also similar in birds like Disco, who is able to imitate sounds of other humans?
Yeah. So birds do have auditory. So they not only learn what we call the acoustic structure of the sounds, but they learn the order in which the sounds are produced. All right, and some species just learn that one sequence, and that's all they do for the rest of their life. Other species, like these budger, like Disco, as you can see, Disco was producing sequences of words or sounds, and sometimes actually making new combinations of sequences that he never learned. So there are some species like us and these parrots that can take these sequences and put them into new combinations. Whether they hear it or haven't heard it before. All right. I'm going to say one more thing before we close out, just something I want you to walk away with. I thought somebody might ask, so I didn't say it early, but I'll say it now, is that I presented to you a view that we have been developing over the last 20 years, and more since I was a student and now a professor. And but this view that I presented to you is not the only one out there. There's some out there, and I just don't want you to walk away with thinking this is the only one. But I think this is the best one, all right? But there's some out there, and you'll see it in this article, where there's this idea, particularly coming from the linguistic community, that there's a language module in the brain. And this language module is controlling everything else, like how to understand speech, how to produce speech, and so forth, telling the speech pathway what to do. So far, we find no evidence of a language module. I think that these brain pathways that are controlling your speech, your language, is really embedded within the brain pathways that are controlling all other behaviors and doing it in a similar way. The second is, you'll hear some theories out there that there's a difference in the vocal organ, that the larynx is more descended in humans compared to other species, allowing us to have greater variety of sounds. there's not any more good evidence for this either. The larynx is actually high or low in different species, different individual humans, and it makes no difference in terms of the number of sounds you can produce. So I just wanted to throw those two ideas out the window for now and finish off. And when we come back, I'll tell you more about the journey that got me here. Thank you very much.